Welcome to the Cell Series Semi-Autonomous Organelles video. At the end of this video, students should be able to describe the semi-autonomous organelle structure and function within a cell, as well as their origin. Semi-autonomous organelles are those which grow and divide on their own. They contain their own DNA and are membrane-bound. These organelles are only semi-autonomous because they still use nutrients from the cell to carry out metabolic processes. Most proteins found in the semi-autonomous organelles are brought in from the cytosol. These organelles are the mitochondria and chloroplasts. Mitochondria are the powerhouse of the cell. Think of them like a battery, creating energy, or ATP, for the cell to use. Mitochondria are bean-shaped organelles that metabolize molecules in the cell by performing cellular respiration, which we will discuss next week. The mitochondria is made up of two membranes, separated by the inner membrane space. The inner membrane forms folds called cristae. The space within the cristae is called the mitochondrial matrix. The outer membrane contains porin channels to filter molecules, not allowing large molecules to cross. The inner membrane only allows certain molecules through. It is highly selective compared to the outer membrane. The inner membrane uses transport proteins specialized to only allow necessary molecules needed into the matrix. Mitochondria contain their own circular DNA and ribosomes, both similar to those found in prokaryotes. Their DNA and ribosomes, along with enzymes, are housed in the matrix. Similar to plant chloroplasts, mitochondria create energy in the form of ATP and metabolites needed by the cell. Mitochondria grow and divide on their own within the cell by binary fission, just as prokaryotes do. In fact, if the mitochondria were removed from the cell, it could not produce any additional mitochondria on its own. Mitochondria are inherited from the maternal lineage, as sperm cells only carry mitochondria in their tails, which falls off after fertilization. On incredibly rare occasions, the sperm tail may be integrated into the newly forming zygote. The rate of change of the mitochondrial DNA, also called mtDNA, is very slow, making mtDNA a very useful tool in the study of human evolution. mtDNA, or mitochondrial DNA, is also used in forensics for identification of intact or pieces of human remains. mtDNA may be to blame for genetic diseases such as Alzheimer's and type 1 diabetes. Endosymbiosis is the idea that a large anaerobic prokaryotic organism engulfed a small prokaryotic cell that was not digested, probably because it was resistant to the digestive enzymes. Over time, the two organisms developed a symbiotic relationship, with the larger organism providing food and nutrients as well as protection for the smaller organism. In turn, the smaller organism began providing the larger cell with ATP and possibly a way to detoxify oxygen. The competing theory states that an archaebacteria engulfed a facultative anaerobe, which is an organism that can live in or out of oxygen. Eventually, over time, this larger organism became what we know now as eukaryotic cell, and the engulfed cell became the semi-autonomous organelles. In mitochondria, it is believed the small prokaryote was a phototrophic purple proteobacteria, as well as in the chloroplasts, a phototrophic cyanobacteria. The signs of endosymbiosis include the double membrane, separated by the inner membrane space, separate circle mitochondrial DNA, which is similar to prokaryotes, the prokaryotic-like ribosomes, and their ability to grow and divide. Finally, mitochondria and chloroplasts have been found to be susceptible to certain classes of antibiotics. Weird fact: today the Paramecium bursaria, a non-photosynthetic pond water ciliate, engulfs green algae and stores them instead of digesting them. The Paramecium pack in so many algae cells that it even looks green. In the light, the photosynthetic algae makes sugars for itself and for the paramecium. And in the dark, the paramecium shares its food with the algae. Chloroplasts are photosynthetic disc-shaped organelles found in the eukaryotic plants of algal cells. Chloroplasts are like organic farms that produce food for the farmer. Chloroplasts harvest sunlight and convert it into chemical energy to form glucose or food usable by the cell. Prokaryotic cyanobacteria, conversely, use phycobilioproteins or phycobilins as their site for photosynthesis, and they do not contain chloroplasts. Chloroplasts are surrounded by a double membrane with an intermembrane space between them. As in mitochondria, the outer membrane is more permeable, allowing nutrients across. The inner membrane is covered in transport proteins, only allowing specific nutrients in. Inside the cell membrane is a semifluid. It's a fluid-filled stroma full of dissolved enzymes. Like the mitochondria, chloroplasts contain their own DNA and special ribosomes. The stroma is encapsulated by the inner membrane, which connects to the stroma lamellae. These keep the grana a safe distance from one another to increase efficiency. The grana are formed from stacks of thylakoid discs, which are flattened, fluid-filled compartments created from the lamellae. Each thylakoid disc contains the sunlight-trapping molecule known as chlorophyll. These discs are hollow, with the interior cavity being called the lumen. There are other photosynthetic pigments, carotenoids in carrots, phycocyanin in bacteria, phycoerythrin in algae, and fucoxanthin in brown algae. Chloroplasts, like mitochondria, also grow and divide by binary fission. Plastids are plant cell organelles involved in energy storage and the synthesis of metabolic materials. Leucoplasts are colorless storage of starch, oil, and protein. Amyloplasts are leucoplasts found in tuber and root veggies like potatoes. Yellow, red, and orange-colored chromoplasts manufacture carotenoids, which can be found in carrots, fruits, and veggies like pepper, yellow and red and orange. The green contain chlorophyll, A and B, for light absorption. Protoplastids are undifferentiated cells found in the plant meristem. They contain copies of the same genome and are all differentiated into the appropriate plastid once the cell needs them. Students should be able to explain the theory of endosymbiosis. Students should be able to describe the components and function of mitochondria. Students should be able to indicate how mtDNA is inherited. Students should be able to understand the components and function of chloroplasts. Students should be able to determine what makes organelles semi-autonomous.